In this activity, we're moving from the outside of the root to the inside. You're going to examine a cross-section of a monocot root and identify the major tissues that make root function possible. Just like with stems and leaves, roots are built from familiar tissue systems, but they're organized in a very specific way to control water movement, support transport, and allow new roots to form. Your job here is to learn that layout, recognize each tissue under the microscope, and connect structure to function. Roots share many structural similarities with stems. They have an epidermis on the outside, ground tissue in the form of cortex, and vascular tissues, xylem and phloem, for transport. But roots also include specialized tissues that you don't see in quite the same way in stems, especially the endodermis and the pericycle. These tissues are critical for regulating what enters the vascular system and for forming lateral roots. In many monocot roots, you'll see a central pith made of parenchyma cells, which play a role in storage and internal support. There are two tissues you should pay special attention to in this lab. The pericycle is a meristematic layer located just inside the endodermis. This is where lateral roots originate, so even in a mature root, this tissue is still capable of cell division. The endodermis is the innermost layer of the cortex. Its cells contain the Casparian strip, which is a band of waxy suberin. This structure acts like a checkpoint, regulating what can and cannot enter the vascular system. Together, these two tissues play a huge role in controlling transport and growth in the root. In a monocot root, the center is occupied by a pith made of parenchyma cells. Surrounding that is the vascular cylinder or stele, which contains the primary xylem and primary phloem. Typically, the xylem is located closer to the inner edge of the stele, while the phloem is positioned more toward the outside. Outside the stele, you'll find the endodermis, then the cortex, and finally the epidermis on the outer surface. Many epidermal cells form root hairs, which dramatically increase the surface area for water absorption. This tissue arrangement increases efficiency since the root hairs absorb the water, the cortex stores and moves the water, the endodermis regulates uptake, and the vascular cylinder transports water. Before you look at your slide, pause and think about this question. How might the arrangement of tissues in a root help the plant absorb and transport water more efficiently? Think about the order of tissues from the outside inward and what water has to pass through before it reaches the xylem. Write your prediction in your lab notebook before continuing. We will start by looking at this cross-section of greenbrier on scanning power so you can see the entire cross-section and get oriented. First, find the epidermis. This is the outermost layer of the root and is usually only one cell layer thick. These cells form the surface of the root, and in living roots, many of them would produce root hairs to increase surface area for absorption. In your slide, think about the epidermis as the outer skin of the root. Next, look for the endodermis. This should appear as a distinct ring of cells closer to the center of the root. These cells are often more darkly stained and more tightly packed than the surrounding cortex cells. The endodermis forms a kind of boundary between the cortex and the vascular tissues and plays a major role in regulating what enters the stele. Everything between the epidermis and the endodermis is the cortex. This is a broad region made mostly of parenchyma cells. These cells usually look fairly large, thin-walled, and loosely packed, and they take up a big portion of the root's cross-sectional area, some as large as 75% of the root. The cortex is involved in storage and in movement of water toward the center of the root. We are switching to a higher power as needed to examine the stele more closely. This is where you'll identify the pith, the pericycle, the primary xylem, and the primary phloem. Start at the very center with the pith. In a monocot root, the pith is a central region made of parenchyma cells. These cells are usually fairly large, have thin cell walls, and look more lightly stained compared to the vascular tissues. The pith helps with storage and internal support and gives you a good reference point for where the middle of the root is. Moving outward, look for the primary xylem. These cells are typically larger and have thicker cell walls than most of the surrounding cells. They're found closer to the inner part of the stele and often look more rigid and more heavily stained because of those thick walls that help conduct water and provide structural support. Near the xylem, but closer to the outer edge of the stele, you should find the primary phloem. Phloem cells are usually smaller and have thinner walls than xylem cells. They may look more compressed or less open, and they're involved in transporting sugars rather than water. Just inside the endodermis, you'll see the pericycle. This is a thin, often subtle ring of cells. Even though it can be easy to overlook, it's a really important tissue because this is where lateral roots originate. As you're identifying these tissues, focus on three main clues: where the tissue is located, how large the cells are, and how thick the cell walls appear. Use those features to help you tell each tissue apart and record how you distinguish them in your lab notebook. Now create a labeled drawing of the monocot root cross-section as you see it under low power. Be sure to include and label the epidermis, cortex, endodermis, pericycle, pith, primary xylem, and primary phloem. Focus on showing their relative positions and organization rather than making a perfect artistic drawing. Pause here while you complete your sketch. Now let's think more deeply about what you observed. For the first question about the cortex and pith, focus on location and function. Even though both tissues are made of parenchyma cells, they play different roles in different places. For the second question, compare both structure and function of the epidermis and endodermis, especially in terms of protection and regulation of movement. For the last question, think back to monocot systems and compare their tissue arrangement to what you see here in the root. Write your responses in your lab notebook using your observations to support your answers. To wrap up this activity, monocot roots have a very organized internal structure that includes the epidermis, cortex, endodermis, pericycle, vascular tissues, and a central pith. The endodermis plays a key role in regulating what enters the vascular system, while the pericycle is responsible for producing lateral roots. The arrangement of xylem and phloem supports efficient transport of water, minerals, and sugars. Once again, the big idea holds true. Structure supports function. The way these tissues are arranged is exactly what allows the root to absorb, regulate, and transport materials effectively.